This chart plots the projected local trajectory of type 2 diabetes. By the year 2045, the data points to a staggering 629 million people living with the disease. Type 2 diabetes and obesity represent a direct failure of the human body's biological energy management system. Finding a cure for these metabolic failures requires testing thousands of potential chemical variables and interventions. However, routing every experimental variable through human and mammalian clinical trials is slow, highly expensive, and constrained by ethical limits. To solve a fast-moving global epidemic, medical researchers need a way to safely run thousands of simultaneous experiments on a fully functioning biological energy system. They found their testing ground in Drosophila melanogaster, the common fruit fly. In a modern laboratory setting, this insect operates as a highly advanced scientific tool. A fly's entire life cycle spans just 10 to 12 days. Researchers can observe generational disease progression and test the efficacy of experimental drugs in a matter of weeks rather than decades. To scientists, the fruit fly is a miniaturized, fast-forwarded live-in test tube. This diagram illustrates the deep evolutionary conservation between our two species. Roughly 60% of fly DNA is identical to human DNA, and 75% of genes responsible for human diseases have functional equivalents in the fly. We can map our anatomical structures directly to theirs. The human pancreas functions identically to a cluster of neurons in the fly's brain called insulin-producing cells. Both structures secrete insulin-like signals to manage systemic energy. In humans, the liver and adipose tissue handle the physical storage of that energy. The fly has an equivalent central hub, an abdominal structure appropriately named the fat body, which stores and mobilizes metabolic resources. Humans and flies operate on distinctly different physical hardware, yet run on virtually identical metabolic software. Because the metabolic systems match, researchers can intentionally trigger a system crash in the lab by raising the flies on diets overloaded with either sugar or fat. The fly's physiological collapse perfectly mirrors the onset of human type 2 diabetes. Their internal systems develop severe hyperglycemia and sweeping insulin resistance. The physical symptoms are severe. The insects become highly lethargic and accumulate dangerous levels of excess storage fat, clogging the pathways that usually transport energy. Once the flies are in this crashed state, scientists deploy high-throughput screening. They use thousands of these sick flies to simultaneously test hundreds of herbal compounds, new targeted therapies, and dietary adjustments. During these rapid trials, researchers track specific metabolic circuits, like the FOXO signaling pathway, which regulates cellular metabolism, to pinpoint exactly which experimental drugs successfully force the system to reboot. By breaking the fly's metabolism on purpose, scientists create an accelerated, low-risk environment to isolate and test potential human treatments. This visualization shows a recent genomic victory. Data from large human populations revealed that people carrying a rare mutation in the MAP3K15 gene are up to 35% less likely to develop diabetes, regardless of their body mass. Knowing the protective gene exists is only the first step. Before a drug can be synthesized, scientists have to map the physical mechanics of exactly how that mutation interacts with the energy system. To translate this into a treatment, researchers manipulate the fly's genetic code to match the human mutation. By watching the genetic sequence play out in a living organism like the fruit fly, they isolate the exact mechanism of biological protection. The fly allows scientists to observe static human genetic data in motion, showing precisely how a specific mutation alters internal chemistry to prevent disease. This rapid testing model is expanding outward. Researchers are currently using flies to develop highly specific precision medicines and to quickly identify hidden endocrine-disrupting toxins in our environment. We are facing a massive global health crisis involving hundreds of millions of people. Addressing it requires a return to our shared biological roots. Using the common systems inside this insect, we test interventions that will define future medical care.
Thanks so much for spending some of your time with us today. We build these videos for you, so your feedback is our compass. What's one subject you've been wanting to see a deep dive on? Let us know in the comments section. We read through your suggestions to decide what we film next. Don't forget to subscribe and turn on notifications so you don't miss our next upload. Stay curious, and we'll talk to you in the comments.